or tonight, let's say, tonight we're gonna see how, protecting our forests is closely linked to having our delicious and healthy cup of coffee. So the first thing, sort of talk a little bit about coffee. So the coffee plant, the scientific name is Coffea arabica. It's a woody shrub, and it's in Africa. More or less, we know when the colonizers, the British colonizers went to Africa, they up and they took it to Asia in the early 1700s. And then, it was, of course, taken to European countries. And then the crop, in the European countries to be consumed. And then also the crop sent the Americas, where it's been planted for a long time. So this is a little bit of the history of the movement of coffee. So who consumes coffee? Who are coffee lovers? As you can see in this graph, they're mostly from Europe, North America, and this is the per consumption in 2018. Sample Finland consumes, 10 kilos. This is an average per year, each person. The U.S. is, is money. It consumes, 4 more or less kilograms per capita per year. I definitely drink more than 4 kilograms a year. I'm sure there's a lot of other people that drink a lot. Certainly the coffee consumption has increased in the last few years. And as you can see, these thousands of kilograms, I mean, these are, millions of coffee bags that are consumed, and that it's been increasing. The next thing, well, who produces coffee? Coffee is grown millions of farms worldwide. The interesting thing is that the majority, up to %, are smallholder farms in most developed countries, including some low human countries. For example, Costa Rica, coffee is produced in farms that are between 1 and 3 hectares. So they're really small, and they're usually managed by the same family. So these are family-owned farms, and most of the coffee is planted in that way. Coffee production gives jobs for 125 million people. So it's a lot of jobs. The top coffee producers are, these low-income countries mostly, and the bigger is Brazil, and goes to Vietnam, Indonesia. These are the top producers. But who produces the highest quality coffee, and in a, let's say, sustainable way? When I say sustainable way, means fair trade or low side type of production. Let me check. Does it seem okay? No, I thought my microphone was working. I'm just gonna go back just a second. So this is important because not all the countries are coffee sustainable, and all the coffee is good quality or is good taste. These are the countries that have some of the best quality coffee with some of the sustainability practices are Costa Rica, Kenya, Ethiopia, and there's a couple more, 6 or 7. One important thing that is happening now is that climate change is happening with many other is that it's been affected by change. So what is happening is that the temperatures are increasing, and the temperatures in the evenings. So usually coffee in mountainous areas where the temperature at night is a little lower. If the temperature becomes a little bit higher than normal, then it's pathogens, organisms that cause disease will have a better environment for growing and attacking plants. So that's one thing. And also the temperatures, the increases in temperatures are affecting the droughts or having more droughts. So these droughts and these increases in temperatures also affecting, increasing the susceptibility to the diseases. And of course, famous, or let's say infamous disease is the rust fungus. Let's talk a little bit about that. The loss in coffee production, especially of the rust, but many others that are related to climate change, is I don't know, something is happening with my microphone, but sorry. This is affecting the production, and some plantations in some Central American countries, they have had losses of 100%, meaning that a lot of these producers or these small farmers, low-income families, are abandoning coffee production. So this, as you can see here in this article, there was a few. The loss in just a couple of years has been millions of dollars, thousands of jobs, just in Central. And as you can imagine, also the immigration, illegal immigration of a lot of the people that were escaping these problems. One of the important diseases of coffee is the coffee rust, and you can see these orange dots here, and these are the spores of the fungus. The fungus, the rust moved from Africa, of course, where it's native, and like I said, it followed the coffee. So if first, of course, it's native, and then it moved to Asia, and then it started to other countries where it's planted until it got to the 1970s. In the last decade, it's caused many more problems because people think that maybe the fungus is becoming more severe and it's becoming stronger, but it's not that specific. It's mostly climate change what is making the plant more susceptible to these diseases. Another important disease is the American leaf spot. So you get little spots. And this disease is very important in Central and South America. We have also in other, especially in Africa, the diseases, fluffy berry disease, and here I put the scientific name of that, or the Latin name, like call it Totricum. And there are different species cause different little of them are in Africa, some in Africa. And there are many, many other diseases, some of the most important ones. And there's, of course, there are insect pests, and there's some other that can attack coffee. And like before we started the talk, we're talking about organic, and this is because chemicals are becoming obsolete. So, and why is that? Well, pollution, the contamination of the environment, definitely. And the use of the let's say, extensive use of pesticides increases the genetic resistance, that like the same fungus or the American leaf, for example, the more pesticides, more fungicides you use, these pathogens become more resistant. So you have to apply more fungicides and more pesticides to create new ones. So then the pollution increases more and more. And another thing, we are demanding better coffee, better meat, better produced, or produce good agricultural practices, fair trade or organic, or with a low use of pesticides. So for example, like I said, sustainable practices are the rice. This is the sort of or production, you say, coffee using sustainable standards. So here is curve. And production, which is the one pesticides or this chemical, is going down. So definitely the demand is important in this case. So how can we produce coffee that is pesticide free? Even though this demand for organic coffee increasing, we still don't have the technology. We still don't have those tools or the good tools that say make organic coffee. So it's kind of a slow process. So how can we produce coffee that is pesticide free? So what I'm trying to trying to study is biological control, which is using beneficial living organisms to control unwanted ones. So in this case, I'm gonna use fungi, good fungi, to attack bad fungi. So this is biological control. So this I compare to probiotics. Okay, probiotics are now kind of a trend. It's trendy, where you find it in yogurts and in a lot of natural supplements. A probiotic is a microorganism that is introduced into the body for its beneficial qualities. Usually you have many probiotics. That's why it's in plural, because you have, in this case, billions of bacteria in these yogurts. They are known to help with digestion and immunity. And did you know that a body, let's say a human body, has trillions of living microorganisms in the body? So meaning in your digestive system, your stomach, your intestines, your skin, your mouth, your vaginal area, your feet, everything is just filled with trillions of microorganisms. And some are good and some are not good, of course. But this is more or less the idea that I want to do with my research, but using plants. So let's say if you have a plant and you have the plant cells, there's tons and tons of microorganisms living inside and between all the cells. There's all the fungi, bacteria, and even viruses are living in there. So for example, let's say this is a microscopic view of a plant, of a leaf, and you see the cells of the plants here, and you can see all these hyphae, what is called is the thread-like structures that form fungal bodies, and these are also called hyphae or fungal mycelium. And sort of these blue threads here, these are all fungi, and this is all filling the spaces in between the cells. And also there's also tons of bacteria that we cannot see in this photograph. They are so ubiquitous in the plant tissues, in the plant parts, that let's say you have a leaf. If you had magic and you were able to remove all the plant cells, every plant material that is there, and just keep the fungi or let's say bacteria, you would still be able to see the perfect outline of that leaf. And it's the same thing with the entire tree, with an entire plant. If you remove all those plant cells or the plant tissues, you can still see the perfect outline of that tree because it's completely filled with these microorganisms. So my research is discovering plant probiotics in the natural forests where plants are healthy. The principle of biological control is that you go to the area where the plant is native or where the plant may have these natural probiotics or these beneficial microorganisms living with them and that have evolved and lived with them for millions of years. When you take seeds from a forest or from wherever and you go plant it, in a monoculture plantation, a coffee or corn or whatever, you're not bringing all these beneficial microorganisms that originated with that plant, millions of years ago. So it's a sterile plant. So the idea of my research is looking for these organisms in natural forests. Specifically, I'm looking for beneficial endophytic fungi. So the word endophyte is divided into two, which means which is first endo, that means inside, and then you have phyte, which means plant. So an endophyte is a fungus, or it could be a bacterium, but now we're going to talk about fungi. Fungi that live inside the living plants without causing damage. Okay, so some of these endophytes are beneficial. There are others that we don't know really what they're doing inside of the plants. There are others that later in life, they may become a pathogen and cause a disease on the plant when the tree is weak or something like that. Okay, but now I'm going to focus on beneficial endophytes. Okay. So my research is in Costa Rica, here pointed by the arrow, just in case people confuse it with Puerto Rico, which has definitely happened to me before. So yes, this is Costa Rica. Why Costa Rica? Well, Costa Rica has a lot of coffee relatives. It doesn't have the natural coffee plant, but it has many coffee relatives. I'm working really closely with the coffee industry in Costa Rica with coffee co-ops to really know what their needs are. What are the most important diseases? What are their goals in their productivity? Who are the farmers? And so a lot of these. So I'm working really closely with them, and this has been very rewarding. So what do we do to look for these beneficial microorganisms? So we go to the forest. We collect samples. So in this case, I'm collecting the tissues of the plants that are, like on the stems. So I take like the bark, the living part of the bark, and I put it on Petri dishes with nutrient agar because I want to grow fungi in that agar, okay? So this is, you have the little pieces there. I also take samples of the leaves. So not only the stems of the plants, but also the leaves. I go to the lab. I take the little pieces of the leaves and culture them on, take the cut in little pieces, and then I put it on the Petri dishes and wait to see what things grow. After several days, I have a Petri dish with the fungal mycelium or the hyphae, which are these very fine thread-like structures coming out of the plant tissue. And these are the fungi. So we do this for many, many, many samples. We have, I don't know, hundreds of samples of plants and stems and leaves, and we culture this until we have many, many, many cultures or individuals, let's say, of the fungi that we found from those plants. So what we have, then the next thing that we do let's say we have thousands of these fungi or these fungal individuals, we study their DNA. Because we have thousands of things, we're not gonna test thousands of things in our biological control experiments. They will take us several lifetimes. So the first thing we do is to study their DNA to identify the species. Then once we identify them, we decide which ones we want to use in our experiments. So this is all done through DNA.
Mm-hmm. Can we go back to the previous slide?
Mm-hmm. This one?
Yeah, thank you. So they look so colorful. Can you tell us why?
Yeah, so exactly. So imagine that all of these things are one different species. So there's all many different types of species. So first, the fungal mycelium could be a different color. It could be white, it could be kind of dark brown or black. The spores that they produce can be many different types of colors. They can be green, they can be black, yellow, you know, kind of pinkish. The other thing that the fungi do is that they produce chemicals, like chemical substances that are called secondary metabolites. So they kind of release that into that agar, or the petri dish. And because there are many different types of these chemicals, they're different colors. So here you can see all the different colors that the agar has is because of these chemicals that the fungi are producing. So that's the reason.
Thank you.
Okay. So far, we have a collection of several thousands of individuals, or what we call strains. Sometimes people call them isolates. And we have identified them, and we have hundreds of different species of fungi. I don't know, maybe 500 species of fungi. And as you can imagine, we cannot use all of them. So we just, based on their identification, we start to do our preliminary experiments. So the first thing we do some experiments in the laboratory, and I'm going to explain how we do this. So let's say we have the pathogen, and we grow pathogen, and in this case, let's talk about the American leaf blight, which is caused by this fungus called Mycena citricolor, and I'll show you a little bit more. So let's say we grow the pathogen alone in a petri dish. We measure how much it grew in, let's say, 7 days. Now we're gonna grow the same pathogen here, but against the endophyte. So we have the pathogen and we confront it against the endophyte, or we can say it vice versa. We put the endophyte to fight with the pathogen. We measure now how much the pathogen grew after 7 days when it was grown with the endophyte. Then we compare these measurements. Okay. And here you can guess, well, how much did it reduce? How much the reduction in growth was? It was approximately 70%. So it means here that the endophyte had a very good control against the pathogen. So in this case, of course, we did many experiments, many different endophytes against Mycena, one pathogen. And we had some things that worked very well, like down here at the bottom with a reduction in growth of almost 70%, which is really very good for a biological control agent. It's always difficult to have 100% reduction in growth. Probably a fungicide would do it, but we know the negative effects of that. But biological control with 70% is great. You know, some of the things they sell commercially, they can control about 30 to 40% of the pathogens. So this is actually really good. So here in the right, you can see all the little confrontations, you know. On the right, you can see the pathogen, and on the left, the endophyte, okay? So this is Mycena. The other thing we're doing with the endophytes and the pathogen Mycena is we're studying the volatile chemicals that these endophytes produce and see if they can control the disease. So volatile, like the word says, are these kind of substances that we cannot see, that in sometimes, you know, we are able to smell. So if we smell a flower and, you know, that aroma, that's a volatile chemical. Fungi also produce those, but sometimes we might not be able to even smell it. But so we're using these chemicals against Mycena. So the experiment is, okay, we have Mycena, the pathogen, growing without the volatiles of the endophyte. This is how the hyphae, those thread-like structures look in the microscope when they're healthy. This is how the little mushroom, Mycena is a very small little mushroom. This is how they look when they're healthy. Once you apply the volatiles of this endophyte, in this case, we're using one that is called Daldinia, you see the hyphae or the mycelium looks a little bit weird, a little bit different. We look at it in the microscope and it's kind of burned. And then the little mushrooms are also burned. And if you take one of those burned mushrooms or hyphae and put it on another plate, they don't grow anymore. So these volatiles are actually burning and killing that mycena. We're doing some other experiments against other diseases like this one called Choledotricum, which can cause several problems on the leaves or also on the berries. And as you can see, there are some that have very good reduction in growth, up to 65%.
I was wondering how much of that process, that secondary metabolite that is volatile, do you need to produce that morphological reaction in mycena?
I don't think it's very much, because the experiment that we did was just like putting one petri dish, let's say at the bottom, with the endophyte, and then the petri dish with the mycena on top. So it's just a petri dish, and it kind of covers the plate. And just with that, whatever amount it produces in a week or less, I think less than a week, is enough to kind of burn those hyphae or those fungal structures. In the case of how we would use that to really kill the pathogen in the field, that's something that needs to be researched. That's definitely something that we would have to figure out how to, kind of measure and know how much to apply. And that also does not affect any other organisms. So that's kind of still a ways.
Thank you. We have 2 more questions in the chat.
Yes.
Helen would like to know, what is an endophyte?
Okay. So the endophyte is, I don't know, maybe should I go back to the slide? Can I do that? Okay, so I'm gonna go back to the slide and I'll show it again. Okay. So I'm gonna show you 2 slides. I'm gonna show you this one first. So here, if you make a cut of a leaf of a plant and you look at it in the microscope, with really high magnification, you will see the cells of the plant here. Can you see the arrow that I'm moving?
Yes.
This is the cell, the plant cell. And these blue threads are these fungi that are growing in between the cells. This is called hyphae or mycelium. Okay, so this is the first slide I wanted to show you. Then this was the second one. So an endophyte, like the word says, endo means inside and phyte means plant. So an endophyte is an organism, let's say in this case a fungus, that lives inside the living plants without causing damage. So if you, let's say if you were able to have a special vision and you can see everything that is in between the cells of a plant, you would see all these threads living inside, but it's not causing any problems on the plants. Many of these endophytes are actually good for the plant, and this is what I'm studying. Is it clear?
It is to me. Helen gave a thumbs up.
Okay.
And we have 2 more questions now. How does the Petri dish agar environment compare to the leaf? Are results validated on the leaf?
That's a very good question. Definitely, it's a process, it's a long process. So the first thing we do is to do these experiments in the lab so that we can do a selection, or let's say a pre-selection of things that we're gonna take to the field or to study it on the live plant. Because if we were to test 1,000, fungal individuals that we have on the plants, we would never finish, and we would need thousands and thousands of plants to do this experiment. So the first thing is we do this, what is called in vitro, or experiments in the laboratory. Then, yeah, we have to take this to the plant and see how it reacts, because certainly the agar, the petri dish, does not have all the different chemicals, all the different components that a plant leaf has. Also, the agar has, you're gonna grow only one fungus. In a leaf, like I said, there are many other different types of fungi and bacteria. So how are those fungi and bacteria going to interact with your biological control agent? So that's the next step that we have to do in our research. So this is kind of a slow process. Okay.
We have one last question. Have you tried the endophytes against any human pathogens?
No, I have not. Right now, I sort of study mostly pathogens or plant diseases. I'm a plant pathologist. So for people to study these endophytes against human diseases, then, well, I guess you have to be like a microbiologist or work in a pharmaceutical company or somebody that works in that. But certainly there are mycologists like me or other ones that have worked at some point in what is called bioprospecting, which is looking in nature, looking for fungi or bacteria that can produce chemicals that are used in pharmaceuticals to control diseases, or for some other many antibiotics and all kinds of things. So my research, no, I have not worked with human pathogens, just plant pathogens.
Fantastic. That is all for now. Thank you.
Okay. Okay, so then after we do these preliminary experiments in the laboratory, now we want to take it to the plant. So how do we do this? We have, let's say, coffee seeds. Well, these are not coffee, but this is just an example of what we do. So we have the seeds. We plant the seeds. At the same time that we plant the seeds, we also apply the endophyte, or the fungus that we want to see or want to study. We let the little seedlings grow, germinate and grow. We let the endophyte sort of grow inside that little seedling. Then the same way we cultured before from that plant, we culture again to make sure that that endophyte has colonized or has grown inside of the plant, okay? So that's what we do. So this project with coffee in some ways started not too long ago. When we started collecting, maybe it was about 3 years ago. And so this process has been slow. So let's say this part with the seedlings or with the living plants, which we started about 6 months ago. So we still don't have adult plants. So we're starting to work with the seedlings mostly. The other thing we do is, so we want to control how these seedlings with the endophyte behave compared to seeds or seedlings that have been pre-treated with fungicides. So in coffee production, the conventional what is called, they treat the seeds with the fungicide before planting, so that when they germinate, there are no other things that will rot those little seedlings or those little seeds. Okay, so one of the treatments that we do is with the fungicide, and the other treatment is, the seeds with the endophyte and no fungicide. And then we see what happens. These are the germination beds, what is called in the nurseries, and then we let all the little seeds germinate, and we have all the little squares where we're gonna measure and all of that. Okay. So what are we seeing now with the seedlings? We're seeing that the endophytes reduce very significantly seed and the seedling diseases. So for example, like I said, we have the seedlings that we treated, or the seeds, sorry, that we treated with the endophyte, okay? And all of these names here, Ovata, Caturra, CR95, and Catuai, are the different varieties of coffee. So what we did is, we planted a certain number of seeds, and then we saw how many seeds did not germinate or got sick and rotted, okay? So these first columns here on top are all the seedlings and seeds treated with the endophytes. And these ones here at the bottom are the ones that were treated with the fungicide. I think it's kind of, you can see very well the difference where a lot of seeds, many seeds and seedlings are rotted that were treated with the fungicide compared to the ones that were treated with the endophyte, meaning that the endophyte is controlling really well the organisms or the diseases of the seeds and the seedlings. Okay, so here you can see, very little incidence of the diseases here, and here much more. The other interesting thing that these endophytes are doing is promoting the growth of these plants. We are, of course, going to see this process throughout, the life of these plants until they become adults and start producing the berries. But we, like I said, we're starting with the seedlings. So look at this seedling, the coffee seedling. On the right, or the red circle, are the ones that were treated with the endophyte, and on the left, without the endophyte or with the fungicide. Also, they can promote the growth. I mean, you can see in this experiment that we did was with cilantro. So we applied this endophyte in cilantro, and you can see how much more growth was on the ones that were treated with the endophyte. And with the coffee plants, also you can see the one here on the right, the leaves look really nice and green and much bigger than the one on the left without the endophyte that is kind of yellowish and much smaller. So the endophytes are also promoting growth. So controlling disease, promoting the growth, and also interesting is protecting against drought. So we did this experiment where we had, let's say we had a total of 100 plants, 50 plants. I mean, the 100 plants we did not water for one entire month, not a drop of water in 1 month, 30 days. Half of them had the endophyte, and the other half did not have the endophyte. So after 1 month, the ones that did not have the endophyte, they all died, 100%. They even died like by the 3rd week, they were all dead. We compare it to the ones that were inoculated or that had the endophytes, only 50%. So this is very interesting that these endophytes can also protect the plants against drought. The other thing we're doing is with the coffee industry is kind of making these let's say these endophytes or these fungi accessible to the farmers in a product that they can actually apply to their plantations, because we're not going to give them petri dishes with mycelium. They want something they can spray. That's what they want. So we grow it here on the on the top right. We can grow it on the rice, and you can see all these green things. These are the green spores. Here are the little bags. So they are grown in a very artisanal way. And then you kind of mass produce the spores until you have a product that you can give to the farmers. So this coffee co-op, for example, gives this product for free to the coffee farmers of their co-op. So with this, you know, we can, what we want to see is how, let's say after 1 year, 1.5 years, when the plants are adults and they're gonna start producing the coffee berries, how this is going to improve the productivity, how the plants are gonna look, are they gonna measure, is it, you know, are the berries gonna, you know, be produced much more? So, you know, after that, what we hope is that really, you know, we can increase the productivity and definitely improve the economies of these farmers or the families that depend on this coffee production. And like I said, it will take maybe about a year, yeah, 1.5 years more or less to have this sort of data.
We have a question that is common among two people.
Yes.
Do you have any idea of the mechanism of increased growth? And could it be a mechanical retention of water by the hive?
There are sort of different, certain mechanisms. I still don't understand exactly how it works, because it has to do with, you know, producing certain secondary metabolites. Also how, for example, for promoting the growth or protecting against drought. There are certain chemical signals that are sent throughout the plant. There are also antioxidants. There were several things that the fungus makes, that, how can I say it, that the plant produces because the fungus is there. It's not necessarily because the fungus produces that. It's the reaction of the plant. So it triggers those mechanisms. So it's genetic mechanisms mostly. But it's not retention by the water by the fungus.
Thank you.
Mm-hmm. So yeah, I mean, that's something that we're definitely going to be working on, is knowing these mechanisms. And this is kind of complicated work that we have to do with, you know, studying, you know, how the plant changes the genome and how the chemistry changes once you apply that. And that takes a little bit of time. So what are some of the things we're doing? Well, we're want to do the experiments in adult plants. So now, you know, we've gone through the process of the seedlings, and we have the young seedlings. We have now transferred those seedlings to the bags, those kind of potting bags, where they're gonna keep growing for a few more months. And then in about 3 months, or at the beginning of the beginning of the planting season, which is kind of in April, they will be planted, and then we'll wait for about a year before they become adult and maybe start producing. So we have to do all of these different experiments with the adult plants. And to study the rust fungus, for example, it has to be done on plants that are not quite adults, but almost adults. And they have to be living plants. We cannot do this in the laboratory. So we have to do these experiments with the rust. The rust is one fungus that does not grow on an agar plate. This is what is called an obligate pathogen. So that's why we cannot study it in the lab. The other thing we want to do is use many microorganisms. So as the word said, probiotics, we're talking about many microorganisms, not just one fungus. So we have to do all these experiments, putting all these different fungi together and see how it can help the plant. And then definitely we want to see how it's going to improve or impact the productivity after, you know, a year, 1.5 years. And then after maybe much longer, can it even affect the taste or the flavor of the coffee? Because the fungi, as I told you before, produce chemicals that can give flavors, aromas. The fungi or these endophytes can also make the plant produce chemicals, so it can maybe even change the chemistry of the plant. We don't know. So that's one thing that we have to see, like you can imagine, in 2 years or so. So now coming to the end of my talk, I mean, I'm I hope that now you can see the connection between natural forests, all these microorganisms, and your morning cup of coffee. Of course, there's tons of people I have to acknowledge, collaborators and funding and students. There's tons of other people that have helped me throughout my, projects related to coffee and these endophyte stories. And I'll just leave you with this last quote by E.O. Wilson that says, we should preserve, or let's say treat, every scrap of biodiversity as priceless, while we learn to use it and come to understand what it means to humanity. And that's it. Thank you very much.
Thank you so much. That was fascinating. And we have some questions for you. So Elaine would like to know, is the shade-grown coffee at any increased risk for pathogens?
Actually, what's been shown in research and also in practice actually is that it's less probable to find diseases in shade-grown coffee. That's why most of the coffee, well, at least in certain countries, I know that many don't use shade, but the ones that are shade-grown, they use less chemicals. So the countries that have more sustainable practices, they use shade. The shade, what they do is not only provide shade, but they use plants that let's say have microorganisms associated to them in the roots that help to fix nitrogen. So then you don't have to apply so many fertilizers. And that helps create an environment that is healthier for the coffee plants. So it's actually less likely to find diseases in shade-grown coffee.
Awesome. Helen will like to check back with you in about 2 years to see how this research develops. I think we're all wanting to know what comes out of this.
Yeah.
Could you share with us what the genus of the endophyte that you're using as biocontrol for Mycena is?
With the volatile chemical or the other ones on the confrontation?
The one that you're cultivating to give to the farmers.
Okay. That's the genus Trichoderma.
I remember.
It's a very, very famous fungus, or it's a genus. So it's Trichoderma is the name of the genus, and it has hundreds of species. So, I'm testing one or trying one or a few trichodermas that were from the natural forest that have worked really well. So, yep, trichoderma. I don't know if you want to write it somewhere, but.
Oh yeah, I can write that in the chat. Thank you.
Thank you. Thank you for inviting me. This is great. It's always a challenge to prepare a presentation that is non-scientific. But I hope that today you learned something.
Thank you.
Priscila?